PCR. Polymerase chain reaction, PCR, is a groundbreaking technique in molecular biology that revolutionized DNA amplification. By rapidly copying specific DNA sequences, PCR enables scientists to study and manipulate genetic material with unprecedented precision. In this video, we'll explore the principles behind PCR and discover its wide-ranging applications in research, diagnostics, forensics, and biotechnology. The PCR process involves three main steps. 1. Denaturation. The DNA sample is heated between 94°C-98°C to separate the double-stranded DNA into single strands. 2. Annealing. The temperature is lowered to 55°C, allowing short DNA primers to bind to the complementary template DNA sequence. 3. Extension. The temperature is slightly raised to 72°C, and a DNA polymerase enzyme extends the primers, synthesizing new DNA strands. These steps are repeated for multiple cycles to exponentially amplify the target DNA sequence. Key applications of PCR. 1. Genetic research. PCR allows researchers to amplify and analyze specific DNA sequences of interest for DNA cloning, DNA sequencing, and studying gene expression. 2. Diagnostics. PCR is employed for disease detection, including identifying infectious agents and genetic testing to diagnose genetic disorders and screen for genetic variations associated with diseases. 3. Forensics. PCR helps in forensic investigation by amplifying small amounts of DNA from crime scene samples, DNA profiling, identifying suspects, and linking evidence to individuals. 4. In environmental studies, PCR is used to detect and study microorganisms and pathogens in soil, water, and air. Samples. It aids in understanding ecosystems, identifying environmental contaminants. 5. In biotechnology, PCR is used in recombinant DNA technology to clone genes, produce genetically modified organisms, and develop novel therapeutic agents. 6. Research and development. PCR facilitates the creation of DNA libraries, mutagenesis studies. 7. Paternity testing. PCR-based DNA profiling is used in paternity to determine biological relationships between individuals. Ancient DNA from archaeological and paleontological samples, contributing to our understanding of evolution, migration patterns, and genetic history.